I'm Dr. Dennis Bente, a professor in the Department of Microbiology and Immunology at the University of Texas Medical Branch, which is also the sponsor of the Thought Leaders Lecture Series. Tonight, we're going to hear about another mysterious place, the Earth's oceans. Environments that are extremely important to life on our planet. We'll hear about NASA's plankton, aerosol, cloud, oceans, ecosystems mission, better known as PACE. Through PACE, NASA scientists will study the life and habitats within the oceans to monitor how they interact with our atmosphere. They hope to learn more about plant and food sources in the ocean, the health of our ocean ecosystems, and how those ecosystems may be changing. This important work fascinates me as a scientist and as a veterinarian because I also work at the intersection between animals, humans, and the environment. Recently in Galveston, I've worked with collaborators to test for pathogens in sea life like oysters, which have recently struggled to survive in our Gulf's waters. The mysteries of the oceans are vast as the untold secrets of outer space. Just as our biomedical research aids mankind in more fully understanding human health and wellness, the work of NASA and PACE will lead to new scientific knowledge about the changing ecosystems underwater and how they interact with our atmosphere. I am looking forward to learning more about PACE. I hope you enjoy the session, and on behalf of UTMB, thank you for joining us for this important and enlightening discussion.
Hello, I'm William T. Harris, President and CEO of Space Center Houston. We're a nonprofit and immersive space exploration learning destination that brings people and space closer together. We serve as the official visitor center for NASA Johnson Space Center, focused on sharing the story of human spaceflight with more than 1.3 million annual visitors from around the world. Thank you for joining Space Center Houston's Thought Leader Series, NASA's PACE program, Unveiling Earth's Oceans, presented by the University of Texas Medical Branch. Our Thought Leader Series brings space and science experts from across the country to provide insights and perspectives on space exploration. PACE stands for Plankton, Aerosol, Cloud, Ocean Ecosystem. Experts from NASA Johnson Space Center, partner government agencies, and commercial partners focused on NASA Artemis program for lunar exploration and beyond. Our panelists are subject matter experts who discuss the significance of their research and findings who help us better understand our solar system and future space missions. At Space Center Houston, we believe science learning and space exploration is for everyone, and we're proud to celebrate 30 years as a premier global destination and center of learning. To plan your journey with us, go to spacecenter.org to purchase your time tickets ahead of your visit and review our Know Before You Go guide for additional tips to maximize your fun. And now I introduce our Thought Leader series on NASA's PACE program, Unveiling Earth's Oceans, presented by UTMB. I'm delighted that we're joined by Jeremy Wordle, oceanographer in the Ocean Ecology Laboratory at NASA Goddard Space Flight Center and project scientist for PACE. Dr. Wordle is an oceanographer in the Ocean Ecology Laboratory at NASA Goddard Space Flight Center, where he also serves as the project scientist for PACE. Jeremy decided to pursue a career as a marine scientist in 1988 after returning from an 8th grade science trip to Bermuda Biological Station for research. He joined GSFC in 1999 and has been working there ever since, focusing on improving our understanding of the ocean's biological responses to Earth's changing climate. Specifically, he studies how the spatial distributions of phytoplankton communities evolve over time. Jeremy also has a keen interest in satellite remote sensing, including the on-orbit calibration of ocean color instruments, the development of remote sensing algorithms, and the validation of satellite-derived data products. These, in combination with his subject matter living in a 3-dimensional fluid on a rotating ellipsoid, create a research environment packed with opportunities to contribute to NASA's efforts to better comprehend our home planet. Our second panelist is Cécile Rousseau, who started working at the NASA Goddard Space Flight Center as an oceanographer in 2011. Her research supports and focuses on the role of oceans in the carbon cycle, the effects of climate variability, and trends in the ocean biogeochemical cycle. She's been part of the PACE science team since its formation in 2014 and is currently the PACE science team lead. In addition to Rousseau's role, she's also responsible for the development of the NASA Ocean Biogeochemical Model at the Goddard Space Flight Center. This model involves creating simulated data sets in preparation for the mission and establishing the framework for Earth system models capability to detect the ocean's color data. She's actively involved in the Carbon Monitoring System program, a congressionally mandated initiative aimed at characterizing, quantifying, understanding, and predicting the evolution of global carbon sources and sinks. In her spare time, Cecile participates in various community efforts and panels like the National Academics Committee on Earth Science and Applications from Space. She earned her PhD in environmental engineering from the University of Western Australia in 2010. Our third panelist, Kirk Noblespeace, is an atmospheric scientist working on new ways for satellites to see our home planet and monitoring its response to changing climates. He began his career by studying photography, but evolved to satellite imaging because in many ways, space is the best place to observe the Earth. He ultimately received a PhD in atmospheric science and applied mathematics from Columbia University and continues to capture beautiful images of the Earth's atmosphere, ocean, and land. Dr. Noblespeace also shares his research and passion between two NASA projects, PACE and AOS. PACE, which is currently launched Plankton Aerosol Cloud Ocean Systems missions, where he oversees the validation and accuracy of PACE data, among other tasks. The other is NASA's forthcoming Atmospheric Observing System, or AOS, mission, providing measurements that are critical for clarifying aerosol and cloud processes that drive extreme weather and climate change. Dr. Noblespeace serves as an instrument scientist for one of the sensors in the planned constellation. He has been at NASA Goddard Space Flight Center in Maryland since 2016 and previously spent time at 3 other NASA locations. This is going to be an incredible program, and we're going to first hear from Dr. Jeremy Wordle.
Well, let me start by saying thank you so much for the invitation to be here. It is really, really exciting for the 3 of us to be able to share the successes of the PACE mission and try to get some energy out in the community because it is NASA's next great investment in the combined study of sea and sky and even land. So what we're going to learn from this mission about our home planet is really, really profound. So as of this recording, we are into day 138 of the PACE mission. Now, the reality is this is a 20-year-old mission. In fact, it is legally old enough to drink in the United States. It takes a long time to get something of this size and magnitude on orbit, but we were finally here, and it is really, really terribly exciting, the fact that data are coming down and we can do something with these data. And you'll learn about that as we progress through a series of talks today. So starting at the beginning, what does PACE stand for? It is Plankton, Aerosol, Cloud, Ocean Ecosystem. So you can get the ocean vibe from plankton and ocean ecosystem and the atmosphere vibe from aerosol and cloud. Now we could have added an L for land because we are super capable for terrestrial applications as well. But sadly that came late in the mission and we never really got to that point where acronyms can change. At a highest level, we are a 3-instrument observatory. And what that means is that there are 3 highly capable instruments on the spacecraft that work in tandem for all of the discovery and interoperability that we'll be talking about today. The primary instrument is OCI, or Ocean Color Instrument. It's not just for oceans, it's really much, much more than that. And then we have 2 secondary instruments as well, HARP-2 and SPEX-1. The first is a contribution from the University of Maryland, Baltimore County, and the second is a contribution from a consortium of organizations in the Netherlands. But here at NASA Goddard Space Flight Center, where we are all sitting, the Ocean Color Instrument and the spacecraft were built, and then those other 2 instruments were delivered, and voila, we put the full observatory together. And if you follow that QR code, you can learn a ton about what we're going to be doing with these data. The promise of PACE is to make the invisible visible. And believe it or not, we're going to be able to see microscopic universes from an orbit that is 400 miles above the top of our atmosphere. Now, when I'm talking about the invisible universe, what I'm really alluding to are two different communities. The first are phytoplankton, these brilliant algae that operate like land plants that form the base of the marine food chain. And second, atmospheric aerosols. Now, clouds are a form of aerosols, but let's just focus on aerosols for a second. And what I'm referring to here is basically anything that's not a gas. So pollen, sea spray, dust, volcanic ash. And yes, we can actually see these from space. So let's talk about why NASA gets involved in this, because almost certainly when you hear the word NASA, you're not thinking about earth science. You're definitely not thinking about, say, oceanography, and you're almost certainly not thinking about microscopic algae. That just does not connect from NASA to phytoplankton. So why are we doing this from space, and why do we care? Well, let's get back to phytoplankton and their role here. So the P in PACE is represented on the left here in the ocean ecosystem. The E is represented on the right. Phytoplankton come in all shapes and sizes. As I mentioned, they form the base of the marine food chain. They convert carbon dioxide into cellular material. So they form the foundation of the food web. They taste different though. They sink at different rates. They move carbon around in different ways. And so their distributions and how they're changing as Earth's climate is changing is really, really important to understand. And they change rapidly too. If you don't see phytoplankton today, chances are they might not be there again tomorrow, just the way the whole ecosystem turns around and turns over. So how do we do this from space? Well, they form these massive blooms. So a community can, while small, form such grand numbers and cover such profound distances of real estate that they change the color of what we're looking at. And it's really this color that starts telling us where they are and maybe what's behind the scenes there. So in this case, you're looking at a coccolithophore bloom. It's off the Celtic Sea, so it's in the North Atlantic. And despite these cells being like 10 microns, they form such dense collections that you can visibly with your eyes see these from space. So this is really remarkable that their communities are so large that they become visible not just to our naked eye, but to the satellite's eye as well. As a scientist, I often slip into different kind of vernaculars. So I'm adding this to say, we're talking about phytoplankton, but we might refer to chlorophyll A in the same kind of conversation about phytoplankton. And why we do that is that chlorophyll A is a photosynthetic pigment that is ubiquitous, found in everything that photosynthesizes from land to ocean to inland waters, everything. And it has a very unique absorption signal. And this pigment is the foundation of what allows us to separate a phytoplankton community from everything else. So I'm getting a little bit ahead of myself, but I want to just say apologies in advance because our terminology goes all over the place and we want to be respectful of that. Okay, so I've talked about all of the benefits of phytoplankton, but there are communities out there that are actively trying to harm you. There are some that carry toxins. In this case, you're seeing an image of a cyanobacteria, which is a blue-green algae that contains a neurotoxin. So in Western Lake Erie, you can see, yeah, they form big, massive blooms and mats. The center panel is the water intake for the city of Toledo. So given that there's a neurotoxin in this water, maybe you don't want to be drinking this, showering in it, letting your pets swim in this kind of water. So we don't want to just understand all the beneficial phytoplankton and how they are changing in space and time, but the harmful ones as well. So it's really, really important to have this vantage point of space to understand the evolutions in time and space and all of the different connections with the rest of our environment that change these communities. So let's transition now to the atmosphere. So our search for atmospheric aerosols is very, very similar. We can, from the vantage point of space, see collections of aerosols and their abundance, so to speak. But without instruments like those on PACE, we are still blind to what species of aerosol is there. And you care about this for a lot of different reasons. So again, these are particles suspended in the air, and they can span from pollen to sea spray to urban pollution, Saharan or desert dust, volcanic ash. And they all play different roles in how our atmosphere operates and connects with the ocean and land. So broadly speaking, you have solar or radiant energy beating down on Earth. Some of that energy is reflected. Some of that energy is absorbed. And then other energy, thermal energy, temperature is actually emitted back out into space. So greenhouse gases, and you hear about those a lot in common dialogue and newspapers and so forth, change how much thermal energy is emitted. Where aerosols and also clouds play into all of this is they control how much energy is absorbed. And so you have some aerosols that are absorbing energy, and that is warming our atmosphere. You have others that reflect energy, and that is cooling our air, cooling our atmosphere. And so it's this interplay of various species of aerosols and how they interact with clouds that really form how our atmosphere traps and contains heat. And so knowing the species of these aerosols is really, really important because we want to know the distributions of the ones that absorb versus reflect. They all interact with the ocean and land differently as well too. So this community structure, this invisible universe is really, really important to understand. So the principles of this are very similar for oceans and atmospheres. Like, how do we do this? Well, we are a series of instruments that are passive. We just collect photons. We look at how sunlight interacts with things. So whether you're looking at the ocean or the atmosphere, it's this interaction of different colors of the rainbow that shape how this science progresses. And I'll tell you right now, the instruments we're talking about on board PACE operate almost exactly like your eyes do for the most part. So you have downwelling irradiance, which is a series of happy little photons that'll, in this case, we'll talk about the ocean, make it all the way through the atmosphere, hit the sea surface. And then if they're so lucky to penetrate through the sea surface, what's in the water starts shaping their interaction. So no matter what you're looking at, whether it is the atmosphere, ocean, Coca-Cola, something else, there are only really two fates that photons have. They either get absorbed by the substance and they get transformed into something else like heat, or they get scattered or redirected. And the water in particular absorbs and scatters lights in very unique ways. All water absorbs red light, so that disappears. But the phytoplankton community or other substances in the water will absorb and scatter the blues and greens differently. And that's manifested and shown here in terms of those shapes of those arrows. So if we continue, a lot of that color, maybe those photons get back through the ocean interface and into the atmosphere and then reach the field of view of our instruments. And again, these instruments operate just like your eyes. So the satellite is effectively just collecting different colors of the rainbow, one color at a time. And that is the profound product that we're producing from instrument series like PACE. So this measurement of light intensity is the foundation for how we are seeing this invisible universe. And again, when I say that it's really very similar to how your eyes work, think of it this way. If you're looking at a blue ocean, your brain is telling you, hey, it's probably pretty clear. If you're looking at a green one, it's probably a little bit productive. There's something growing in there. If you're looking at a brown one, then yeah, there's tea or suspended sediments or dissolved organic material there. And this is really just the way the satellite instrument system works. It takes these measurements of color and interprets them just like your eye does, but it does it with sophisticated math and big data volumes, and Kirk will talk about that in a second. So where PACE comes in now is bringing more color to the science community. So what this panel on the left is showing is what colors of the rainbow our heritage missions have launched. And we've been have observed and collected, and we've been doing this for about 40 years. But you'll notice most of them are looking at two different blues, maybe one or two different greens, and just a couple of yellows. When we move to PACE, we're making a move to spectroscopy. And what I mean by that is now we're suddenly collecting all of the colors of the rainbow, from the purples and the ultraviolet all the way to the near infrared. Well, why does that change? Why does that matter? Why is that important to you? Well, if you're walking through a forest, you might simply observed that there are many, many different shades of green, and your brain is telling you that those different shades of green are related to different plants or different species or different health of that species. So measuring only one green might be really great to say, hey, I see there are phytoplankton there, but it's not telling you what community is actually there. So let me give you an example of that. In this case, let's move to the north part of the Arabian Sea and the Indian Ocean here. There is a very profound and important community shift from one phytoplankton that forms the base of the food chain for a very particular kind of fishery to another that has recently come in and started to outcompete it. And it's created a completely different food chain. It's changing the carbon cycle, the oxygen availability in water, doing a lot of different things differently. So this has a lot of meaning for this ecosystem. And we want to observe it from space because we can't be everywhere all times. So if you're talking about the heritage instruments that NASA's been flying for 40 years with partners, when they only measure a couple of colors of the rainbow, or maybe a box of crayons, you could think of it that way, with only 8 different crayons in it, there's not a lot of information to separate these communities. So if you're looking at the middle plot on the far right that says what VIIRS sees, your x-axis is just color, wavelength. So 350, 400, those are the purples, 400 to 450, those are the blues. The reds are 650 to 700. And then the y-axis here is just a proxy for what the satellite would see. And those two different communities are shown by dots that have different colors. The original base of the food chain, the diatoms, the fish food, are in black. The turtle food, the big bully that's coming in, that's Noctiluca, are in red. And you can see in that plot that there's only one unique piece of information. So we really can't tell those two communities apart. But when you have an instrument like the Ocean Color instrument on PACE, what you see on that bottom right-hand corner plot is that full color spectrum reveals all of the differences in how these two communities interact with light. And we can see that from space, and with this information, we can now start telling these communities apart. So before I hand this over to Cecile and Kirk, who will wow you with the amazing data we're collecting, let's just talk very quickly about how these satellites operate. In this case, you can see we are what's called a polar orbiter. We are going from the South Pole to the North Pole, and we see the world below us at 1 o'clock every day, all day. Our first instrument, OCI, this is the primary instrument, is the one that's going to deliver all of the color, and you can see that it has a very nice wide swath here, so we can see a huge parcel of land, and it does this orbit 14.5 times a day, so we're going to be painting these beautiful swaths here. HARP-2, which is one of the two polarimeters we were talking about, complements this swath really well, and it does so in a very interesting way. It looks at the world through multiple view angles. So you're not just seeing your real estate from one different perspective. And the beauty of both HARP-2 and SPEX-1 here too is they also view the world with polarization, sunglasses on, so to speak. So it's a completely unique way that Kirk will elaborate on, I hope, of viewing this universe. And so the information content, by just looking at color in certain intensities and now looking at polarization state, is such a quantum leap forward in information content that this new view provides all of the information we're going to need to start making new discoveries about what we're looking at in our home planet. So hopefully you're reading all this amazing text behind the scenes here. But to wrap up, this 3-instrument synergy is light years beyond anything we've had before. It's filling a substantial niche. We have ocean color observations that I've been talking about in aerosol observations, plus this opportunity with these two multi-angle polarimeters to do cloud and aerosol retrievals beyond anything we've been able to do before. So now that you know everything about how the observatory operates, let's move on to Cecile, and you'll learn about the data products and how it fits into the Earth system ecosystem. So with that, I will leave this up for 1 second as we transition to Cecile, and just say one more time, we're filling some major niches here in our ability to see our home planet, and we couldn't be more excited to be sharing this mission with the world.
Thank you, Dr. Wordle. That was absolutely fascinating. Now we'll hear from Dr. Cecile Rousseau.
Okay, now that we know from Jeremy what PACE is going to provide and why it's so important, let's move on to how do we make sense of the PACE data. And we know that PACE is observing the color of the water. And while it's interesting to know whether a parcel of water is brown versus green or even red, it's also not, what makes most sense to the majority of people, and that is not a really usable type of data. So how do we go from this to the type of needs that Jeremy presented, such as knowing what kind of phytoplankton do we have? We know that some of this phytoplankton is very good to us, and then we know some of them are actually toxic. We also know that in some cases we need to know where to treat water in a different way, or where we shouldn't swim, etc. So we need to really take that color of the ocean into a usable format for our needs on an everyday basis. Similarly to the ocean, in the atmosphere, we have the aerosols and clouds, and these affect air quality as well as the weather and have a direct interaction with the ocean, right? And so we have these two polarimeters on board of PACE that are providing that other piece of the puzzle, because the PACE mission is really an Earth system mission, which means that it's not just one component or the other one. It really thrives to look at the planet as a whole. So if we now dive down into the actual product that PACE will deliver, we will have a range of product within the atmosphere and another range of product within the land and the ocean. Going into the atmospheric product, you will see here that we have aerosols absorption and concentration, as well as a variety of cloud properties. And this is critical because the size of the aerosols, but also the density of clouds, etc., have an impact on what we can see. Like you can imagine if you had a dust storm or a fire, you would want to know what the visibility would be. But you also have a direct effect on the heat that comes to the surface of our planet. And that's important not only for us, but also for the ocean and the organisms that live in the water. Not only does it affect the heat that gets there, but it affects the light. And so if we have more or less light that gets to the surface of the ocean, that is going to dictate quite a bit in terms of production, that primary production, that phytoplankton that not only fuels high trophic level, can be toxic, but also generates over 50% of the oxygen on Earth. There is some product that we know will deliver, and then there's some product going forward that we strongly believe that are going to be possible because of the nature of the PACE data. Something like oil slick detection is going to be a brand new area of product that we are hoping to deliver, and that's going to be based on all that information that we have on the color of the ocean. Now, moving on from the atmosphere into the ocean, as Jeremy showed, the ocean is going to have several types of product, including the phytoplankton contribution and the communities, which means we're going to know how much carbon is taken up from the atmosphere and gone into the ocean to feed higher trophic level, but also to sink to the deep ocean, which is really one of those variables that we really want to know what the future of the global carbon cycle on Earth is going to be. You can also see some of the land variables that we're delivering for PACE, such as land albedo and vegetation indices. This just means how much light comes back from land, and also what kind of vegetation we have. And then some of the other variables are going to be related to the light in the ocean. You might wonder why we care about light. This is very important because it drives most of the production underneath the water and how deep that can go. And I will show one type of application with Earth system models and how it's critical for them to know that kind of data that PACE is delivering to kind of expand the type of application that we can have with PACE. So now we move on to the real data of PACE, and you can see that some of the PACE data here shown have a lot of clouds, and of course we have to remove those clouds. And once we remove that and we get to the surface of the ocean, we see what we call total chlorophyll of phytoplankton, and this is what historical missions have provided so far. So you can see that in yellow you have a lot of phytoplankton and blue not so much. And PACE is pushing that boundary because now we can see what proportion of that phytoplankton is due to picoeukaryotes versus Synechococcus or Prococcus. And being able to differentiate it between those types is telling us a lot about the ecosystem. Now, moving on to HARP and SPEX1, which are the two polarimeter, these provide information on the aerosols and clouds in the atmosphere. And for example, HARP2 here, you can see identifies smoke on the East Coast, and then SPEX1 is really complementary and provides data on the size of the aerosols. So, each of the instrument on PACE is really providing one more piece of information that builds on the next one to really give that overview of the Earth as a system rather than looking at just the water or just the atmosphere or one component. Let's look some more at some of those aerosols and clouds properties that are derived from PACE. And here you see two regions, on the left, Japan, and on the right, Ethiopia. And this is what the polarimeter are telling us. And this is where it gets really cool when we can combine instrument. You can see that at the bottom, both regions show very high concentration of fine particles and somewhat lower coarse particles. But then on the right, you see that in Japan, the absorption of aerosol is very low. But in Ethiopia, it's very high. So that, for example, gives us the question, why would they be different if the fine and coarse proportion is the same? But this is where PACE delivers really cool data because it tells us, by providing us the absorption, that in Ethiopia, the aerosols are actually due to mostly dust, whereas in Japan, it's related to pollution. And if we did not have PACE, we would not have the answer to that. And so this is a very good example of where the combination of instrument and also having that on top of the ocean signal is really help us understanding what's going on in the atmosphere and how that might impact the ocean. I made this picture because I figured, there's nothing better than looking in your own backyard what PACE sees. And this is an image from sometimes in April, I believe. And it really highlights all the spatial variability that you can have in just total chlorophyll. So, we talk about phytoplankton composition, but just looking spatially, you can see on the top right, you have some eddies with lots of phytoplankton, and then you have the coast and you have lots of phytoplankton in the coast. But it turns out that sometimes that high phytoplankton is due to nutrients that, sometimes are natural from rivers. Sometimes, humans, just anthropogenic pressure increases the nutrients in coastal waters, which drives more phytoplankton. And then sometimes it can be due to upwelling. And those questions are very hard to answer. And I'm going to show you next how we take those PACE data and we move them into Earth system model to answer that question. But before I get to that, let's move on to the next slide. And, we talked about how PACE doesn't just see total chlorophyll. It looks at chlorophyll and then it splits it up. And this is one image that shows you on the left what you would see with your own eye if you were flying over that part of the ocean. And then on the right side, you see the total chlorophyll, so what the previous satellite we had would see. And in the middle, you see what PACE is providing. PACE is really giving us an information about which phytoplankton groups are where. And that tells us, are they going to be providing more carbon dioxide sink? Are they going to feed higher trophic level? Can we expect more fishes in that area? And of course, this is just an example with two phytoplankton groups, but there's going to be a multitude of them. And we hope to have some algorithm that allows us for the harmful algal blooms, for example. So we'll be able to map like that where those toxic phytoplanktons are. And that's really, the added value of PACE here in terms of ocean community. So when we move from this and we take those amazing PACE data and we plug them into Earth system model, Earth system models are used because it fills some of the gaps that you can have from satellite data. And what you see here is an Earth system model from NASA Goddard Space Flight Center called GEOS. And it simulates all the aerosols product in the atmosphere that includes dust, but also smoke and sea salt. And you can see the Saharan desert that is blowing dust all across the world. And you can see the actual spatial impact of that. You know, this is not a local problem. This goes everywhere around the world, in the ocean, over land. And then you can see those typhoons on this video that are being generated in the Pacific Ocean, and that lifts up that sea salt. And, you know, you can use models like that to assess the extent and the development of these processes. And then one more thing that is shown here is the fires, the effect of fire. And you're going to see here in South Australia, a fire is developing, and you can start seeing that smoke that is coming out in green. And you see that the extent that that smoke is going to go is way past New Zealand. It gets actually carried away in the Southern Ocean along the water, and that potentially can create effect on phytoplankton. But you can imagine if it was in the Northern Hemisphere, where there was a lot more land mass, it could directly affect, you know, air quality. Now, we also have ocean biogeochemical model, and this is a model by MIT that simulates the different phytoplankton groups. So a little bit like PACE, but on a modeling framework. And the cool thing about this is that these just simulate where you expect phytoplankton to be based on, you know, what we know of how phytoplankton live. But now that we have PACE data, we're actually going to be able for the first time to look at this model data and see if they're actually right or not. And then we can use that to combine both data set and start investigating things like what are driving these things, because the models provide data that the algorithm sometimes cannot provide. If you don't have an algorithm for your satellite for nutrients, for example, your model can provide you the nutrients that are actually happening at that same time. And you can answer some scientific question like, what is driving, you know, this species to bloom in that area? Or what's the effect of a volcano on the phytoplankton? And you can do that forecast at small scale, or you can go, you know, at 100 years. What would happen if we had more volcanoes, or if we had more fires? What would our planet look like? And so that's where, you know, combining those brand new PACE data with Earth system model can really expand the amount of application that we have for the PACE data. And if you want to learn more about the mission, we have a QR code here, both about the mission, but also about the community of practice, which is one particular aspect of the PACE mission, is that we got the application community involved even before we launched, so that we were sure that we were producing data that were relevant to their needs.
Thank you, Dr. Rousseau. And now we'll hear from our final speaker, Dr. Kurt Noblespeece.
So great, thank you for having me. I'd like to build on what Jeremy and Cecile talked about by giving sort of a picture of the day-to-day aspect of what we do with PACE data and how we get to science. So the analogy that I'd like to use here is a factory, a data factory. And you know, many people were involved in the creation of the PACE mission, building the instruments, putting the spacecraft into orbit and all of that. There's still a large team, now that that's been done, there's still a large team of people who are working with the data from PACE. So I'm going to talk about that process now in a little bit. So every day, PACE sends down about 310 gigabytes of data to Earth. These are raw data. These are just the observables, the engineering units that are sent down from all three instruments. And from these raw data, scientific products are generated. And we don't just generate them, we need to test them, we need to distribute them, we need to archive them, and we need to make sure that the larger scientific and I guess general public is able to use the data for their purposes. So like I said, the analogy that I'm going to use is a data factory. We really take raw data, sort of like raw material is, in our case, the data from the satellite, and this is processed into what we call products. And so these are parameters such as chlorophyll A concentration or aerosol, the amount of aerosols And these are scientifically valid products, and we use our factory to go from one to the other. We also have a place where we need to put the data. We need a warehouse or an archive, and we need a way to distribute the data online or in some other means to the general public. So inside this data, there's sort of several steps that we go through. So one, we might start with raw satellite data, and we need to turn that into essentially images, calibrated, geo-registered. That means that we've identified where we're looking with the satellite very exactly, and turn that into something that is, like what the satellite is seeing at the top of the atmosphere from orbit. Next, we need to turn this into scientific data products. What I'm showing here in this slide is chlorophyll A concentration. So that's not something that we don't see that in the same way, but it's a number that has scientific relevance. Now, when the satellite observes, it makes an observation at one time in place, but it can't see everywhere all the time. We're not seeing at night. If there's clouds, we don't see the ocean. Some places there aren't clouds. And so there's value in looking at long-term trends, long-term changes. One of the things that we're doing from space is it's a very unique vantage point to observe our planet Earth, and it's able to perceive things that are sort of beyond scales of human perception, long-term trends, global observations. So a very important part of this as well is aggregating data over periods of time, looking at, say, monthly averages, for example, and then subsequently using this information, combining it with models or using it to inform models and other applications that are of value to everybody. So what sort of jobs that we might have within this factory, and it really does still fit this analogy. So we have people making sure the assembly line works. We have computer scientists, computer engineers, people making sure that the bits coming into the system produce products at the end. There's people who care about or whose job it is to manage the warehouse, in other words, our archive, to manage distribution, how the data are released to the community. And it's done in many different ways, depending on different levels of users, different types of users. We have people who really understand the PACE spacecraft and instrument well. And so these are people who can help us calibrate the instrument, ensure the satellite's working properly, send commands to the satellite to make sure it stays healthy on orbit. We, of course, have lots of scientists here working with the data. We have earth scientists, computer scientists, and these are people who are maintaining specific what we call product lines or generating new products from PACE observations. One of the interesting things about having a new mission with new capability is that we don't know everything we are going to do with the mission yet. We have new products that we need to create and test, make algorithms for, and so these people do that. We also have specialists who communicate results and respond to user feedback. And then we have a large number of people, and this is very important, technicians, scientists, and others who gather data and perform quality control. So quality control is what I'm going to go into a little bit more depth about right now, because it's important not just to have data products, but to know how accurate those data products are. And that really is important if you want to use this for any sort of scientifically valid application. So here's an example of that, and this is from another ocean color observing instrument. This is from an instrument called MODIS, and this is a time series starting in 2003, going 20 years to 2023. And this is a metric for, I guess, sort of average concentration of chlorophyll A pigment in the oceans. And one of the things we want to know is this changing? Is our planet changing, and in what way? And so I'm not going to really talk about whether it is or isn't, but one could look at a figure like this and say, well, if we want to make this sort of assessment, we need to know how accurate the measurements that go into this are in the first place. And that's what these little error bars, which are very small in this, are relevant to. So satellites provide global long-term measurements that help us see trends, among other things, but that's one important role of it. And so we need to know data product accuracy to use them properly. So what the term that we use for this is validation. So what we do is we need to make alternative measurements and compare them to our satellite observations. And we do this by collecting lots of data. So there's a couple of ways of doing this. One way we can do this is to compare to other satellites. And so this is a comparison not with PACE, but between two other ocean color instruments of, again, chlorophyll concentration. And there's different ways you can make these comparisons, and that tell us something about both the conditions under which they're making their observations and the consistency of the measurements. Now, this is useful in some contexts, but you don't always have coincident measurements between two different satellites. And in the case of the PACE, we're making observations, new products, for which there isn't something to compare to from another satellite in orbit. So in these cases, you need to make field measurements as well. And this is an important part of all missions. And so I've just kind of put a couple of pictures on here, and I'll talk through them a little bit. So one way of making field measurements is simply that. You go into the field, you, in this case, maybe put a net into the ocean and measure phytoplankton that are in the ocean. Maybe you take samples of the water or samples of other things. You might take this back to a lab and do further analysis on this. And the example here is for the ocean, but we are also trying to do this with the atmosphere as well. It can be a little bit different in the atmosphere. And so the ways that you would do this, instead of being on a boat, you might be in an airplane. And so I'm showing a couple of images at the right of airplanes that we might fly. Sometimes we might want to fly up real high and look down on the Earth, just like the satellite does. And so we have sort of, we make a satellite that's similar to PACE and we put it on an airplane. In other cases, such as a smaller plane at the bottom, we might fly through a bunch of aerosols or fly through a cloud, and that aircraft has ways of gathering air as it's flying through those things and doing an analysis on that to understand what's there. We have other sorts of instruments on the ground. So at the top right, there's a buoy in the ocean, or a float rather. And at the very bottom right, there's sort of a robotic instrument on the ground that looks at how the brightness of the sun changes as it goes over the sky. And that change in the brightness of the sun tells us something about what's in the atmosphere as well. So all of these things, it's a very sort of complex process. There's many different ways you gather data for validation, and it's very product specific. So for example, for the PACE mission, we are creating a dedicated field campaign. And the idea here is to combine different types of observations and make sort of concentrated observations in one time and place. So this is called PACE PAX. PAX stands for Post-Launch Airborne Experiment. And what we're doing is a collaboration with different government agencies and internationally to make observations that we can use to validate PACE. For example, the top right image is a really interesting airplane called the NASA ER-2. It is essentially a civilian version of the U-2 spy plane. And the nice thing about this aircraft is it flies very, very high. It's about as close as we can get to simulating what observing the Earth from space would be like from an aircraft. And so the nice thing about that is we can put instruments on there, some of which are very similar, proxies for what's in orbit on PACE, and we can fly this at the same time and place that PACE is flying over. And we can do that while other validation observations are being made either from the aircraft at the top left, which is a Twin Otter, a smaller plane that is owned and operated by the Navy Postgraduate School in Monterey, California. And so they'll be making measurements in the atmosphere and the ocean. Bottom left, we have a NOAA ship that will be operating in Santa Barbara, and they'll be making measurements both of the atmosphere and in the ocean. So this is a whole bunch of stuff that we're putting together to make really concentrated measurements in one location and time. It's September of 2024. But of course, that's limited to just this time and place of the world, and we're not going to see everything everywhere like the satellite does. So there's actually a larger program of validation that exists at NASA. These are NASA-funded teams. Some of them are internal to NASA. Some of them are in academic institutions or other places. And so they're going to be going out and making other types of measurements in other parts of the world. The map at the top left shows some of the locations where those measurements were made. Circles might be an area of concentrated focus. Lines might be, say, a ship cruise that goes in those directions. And at the bottom right, there's a table of a bunch of different types of products that will be observed during these field measurements and compared to PACE. So like our factory, we have a process we go through in using validation data. It's not just about collecting the data, it's about collecting the data, make sure it is consistently collected, that we understand its own uncertainties, its own accuracies, and then put this in a place that's usable. So we have a system within our laboratory here where we ingest all of that data, we put it into what we call a QA/QC system, and it's set up so that these measurements can then be connected to the satellite observations and make sort of statistical distributions of how well they compare. And all of this information, which tells us the validation quality, essentially feeds back into our factory again. It might tell us if something is not working the way we expect it to. It might tell us if we have a new product, if it is something worth producing and sort of worth distributing or not, among many other things. The website for this is here. It's not just used by us. You know, this is NASA. It's publicly available, and many other places can use these data as well. And so I think that's all I have to say, really. There's a lot of people that go into making a mission like this work. And once it's on orbit, there's a lot of people maintaining this over the years. Thank you.
Thank you, Kirk. And now we'll transition into our panel discussion. I'm going to ask several questions, and please feel free to participate and join in and help your colleagues answer any of these questions if you like. Our first question is, how do you anticipate PACE's data will help our understanding of the ocean-atmosphere carbon exchange, and what potential surprises do you foresee arising?
Well, you cannot understand the ocean without understanding the atmosphere. And there are a lot of very deep connections that we know about on local scales, but don't entirely understand with a global picture here. And just giving you an example, we're starting to see events where there are cause and effect type things happening. So an example of this would be there were wildfires in Australia in the 2018 timeframe that went way offshore. And those wildfires, created carbon dioxide in the atmosphere, but then deposited iron and nutrients in the ocean. And the response of the ocean was to have a massive phytoplankton bloom. And there are studies now that are talking about how the carbon dioxide conversion into cellular material related to that bloom started to have an offset to the carbon dioxide that was actually created by the forest fires. So not advocating for anything, but it's connections like these that a mission like PACE can start to unravel. And we know now that, hey, deposition of nutrients under the ocean can seed a bloom. We know also that there are certain compounds that phytoplankton release that can seed aerosols and cloud condensation nuclei. We know that carbon is getting exchanged just in the form of photosynthesis. So these new observations from PACE will shed light on this. And the challenges are all related to discovery. We don't know what we're going to find. That's what's really exciting for someone like myself here. So I almost don't want to comment on what I think we're going to find. I want to leave it to the early career discovery scientists out there. There's going to be a ton.
So I'd like to add in a little bit. PACE is a multi-instrument mission, and it's a multidisciplinary mission. And so if you want to understand the relationship between the atmosphere and the ocean, the atmosphere and land, all these things, it's really great in this mission that we have the ability to combine data from different sort of more specialized instruments into a whole package that helps us understand the global system. So that synergy is so important to what we're trying to do.
We often hear about aerosols, and everyone associates them with propellants. But this really provides us with a whole new understanding or definition of what aerosols mean. So can you talk a little bit more about the key mechanisms that aerosols can play in influencing phytoplankton growth?
So phytoplankton, like land plants, grow well when certain conditions are met. And typically this comes in the form of not just sunlight availability, the right temperature, but also in nutrient availability. And so aerosols can deposit key nutrients on the surface of the ocean. And if those nutrients like iron are otherwise missing from that ecosystem, something as simple as that deposition can stimulate a phytoplankton bloom, which is really kind of cool.
That's absolutely fascinating. So I'm understanding that there are ways that we can influence phytoplankton growth by manipulating potentially aerosols. Does that sound accurate, or are there ways that we could influence phytoplankton growth by manipulating aerosols? Is that possible? Could that happen through geoengineering or through some other way of influencing carbon growth?
There has the conversation about like this kind of geoengineering has been coming and going fairly cyclically over the past couple of decades. In fact, there's a famous quote from the 80s. I think it's George Martin who said, "If you give me a tanker of iron, I'll deliver you an ice age." And I might be bungling that a little bit, but the principle was, hey, if we seed these patches of ocean deserts with iron, then you can stimulate a phytoplankton bloom. That phytoplankton bloom will remove carbon dioxide from the atmosphere and sequester it in the ocean. And of course, there's been a bunch of follow-up modeling studies that have supported or refuted all of this. But yeah, to your question about that, this has come up a lot. And in fact, there's a marine carbon dioxide removal effort that is getting a lot of attention right now that touches on some of this. And then of course you can do the same thing in the atmosphere doing other things.
It's amazing the predictive capabilities of PACE. And I'm wondering how you envision the data from PACE transforming our ability to predict, monitor, and possibly even mitigate harmful algae blooms. We hear about these blooms around the world and how toxic they can be, not only to marine life, but also to humans. What are some of the biggest challenges in this area?
So PACE is going to have algorithms that are going to be developed to predict that kind of harmful algae bloom that we observed in many areas of the world. The thing is that different regions have different species, and so the number of algorithms that we'll have down the line is still unknown. But in terms of predicting them, you fall into having some kind of model. It can be statistical model, so you can be like, hey, those harmful algae blooms develop when there's very high temperature, or it can be dynamical, which is where you have those Earth system model that predict. Now, generally, those harmful algae blooms develop in coastal areas, so we're talking the challenge, I think, is going to be very much related to the spatial and temporal extent and somewhat mismatch between how those events develop versus how we are used to model them. They happen at very small scale and fairly rapidly. And models temporally can match that, but spatially, models tend to be much coarser. And so we are at that boundary where we are trying to push it to get to smaller spatial scale to be able to forecast more like a weather forecast, but for harmful algae bloom. But that's a challenge that is mostly related to, computational power and how many processes can we run at the same time to do those forecasts. And I think, if you do this in an algorithm way, so where you just take the satellite data and try to forecast based on that, I think it's going to be the community is going to look at a lot of data and collect hopefully a lot of in situ data and combine them to develop algorithms that are going to hopefully predict many harmful algae bloom to come.
Well, we all are dealing with climate change in different parts of the world, and I can see where the data collected by PACE could potentially help us with predictions. Do you think that the current climate models will be adjusted and be able to integrate the PACE information? What are some of your predictions about how this could influence future climate scenarios?
So So PACE is studying aerosols in clouds, and if you look at climate modeling efforts, one of the largest sources of uncertainty in those models is a relationship between aerosols and clouds. It's actually quite interesting. You know, cloud droplets just don't form spontaneously in the atmosphere. They like to form on something. And that something is often an aerosol, some sort of particle of something in the atmosphere. So if you change the nature and the distribution of aerosols in the atmosphere, you change the type of clouds that are there. And if you change the type of clouds that are there, you change, as Jeremy showed in earlier today, the energy balance of the Earth. So greenhouse gases are affecting, long-lived greenhouse gases are affecting our planet in a certain way, but something else that we don't understand quite as well is the role of clouds and aerosols. So with PACE, we're hoping to better understand that role. And part of the limitations of models has been that they're information limited. You know, traditionally we're able to understand how much aerosols are there, but not so much about what kind of aerosols they are. And so with PACE, we'll be better understanding what kind of aerosols, and that information sort of globally, statistically, at large scales, helps climate modelers build better models. And that's really where it comes into play. And better models specifically related to the role of aerosols in clouds is one way that PACE observations can be very helpful.
Yeah, and I can complement that with the point of view on the ocean. And I think, you know, Kirk is right in the modeling is only as good as how we understand the process, right? So you can't model something that you don't understand. And so in some cases in the model, we have to make assumptions about how things go. And, you know, if I look into the ocean biogeochemistry and a model, there's a lot of things that we think it makes sense, but we haven't had the data up to now to really confirm that. And personally, I'm very excited to see a little bit how we are doing in terms of real data and what was our model right, or are we missing some processes? And this can be like depending on the area. We might be missing some processes in the model for a specific region, or we might be right. So I'm really looking forward to seeing you know, a little bit of that and adding products to what PACE can deliver to kind of help close that loop of system science questions that we have.
Wow, that is so interesting. I can actually see how PACE data could be very valuable in many other fields in helping us understand Earth and so many facets of our lives. Do you see this data being used outside of oceanography and atmospheric science, for example, with public health or agriculture or in urban planning?
So the PACE data are going to be used, and you know, some of the ocean color data are already used for many, you know, application in terms of policy making, but also health-wise. You know, we have some products that are used to assess those harmful algae blooms in lakes around the US, for example. And down the line with PACE specifically, we really have tried from way before the mission launch to make sure that that application community was on board and also up to speed with everything that was going on, to try to really increase that amount of interactions we have and the use of those data from PACE for their specific needs. So that's something that we put a lot of energy from very early on, like 4 or 5 years before the mission launched. We started talking to the end users and, you know, the community that were outside of the science community.
I also wanted to add, and I hope this is obvious, but perhaps it isn't. All of the data that NASA is collecting with PACE are free and available to everybody. And so we're not a policy-making agency. We don't make recommendations about what laws should exist or what regulations should exist. We collect the data and we make it available for everybody. So part of this also is, you know, PACE data are very valuable, but perhaps even more valuable if they're combined with other sources of information, or are there specific knowledge? So for example, one thing that people might care about is air quality. PACE, because air quality is related to the particles in the atmosphere, aerosols, for example. And knowing what we know from PACE is one piece of the puzzle. But if you really want to understand, the specific situation at the nose level in a city, you need to combine PACE data with other sources of information. And so one part of what we do is just make the data available so that it could be combined and provide a value to many others. And I'm giving the example just of air quality, but there's other situations in both ocean, atmosphere, land surfaces, and other disciplines.
We're getting close to the end of our time together, and I've been fascinated to have so many more questions, but I always like to ask, how did you get into your current positions, where you are today? We have many listeners who are really intrigued to understand, how does someone become a scientist doing the kind of work that the three of you are doing?
I resolved to become a marine scientist in some capacity in 8th grade. I was lucky enough to visit the coast and see a research center, and that just that set everything in motion. And everything else is the stars just aligning. Like, I'm really, really blessed. And a lot of that has just been, a cold call to NASA Goddard, where I've been working for 25 years. I've had great bosses. NASA is a really dynamic and energizing place to be, and for a lot of reasons, just never wanted to leave, and opportunities have presented themselves, and the stars really just aligned for me. It's been pretty magical.
For me, it was not actually like Jeremy, where I knew from the beginning I wanted to be an oceanographer. I was always interested in science, but I didn't really think of oceanography being where I was going to end, and certainly not NASA. But one thing led to another, and I did, undergrad in biology, and then, I got focused on oceanography, and then I went to Australia and did my PhD there. And I wrote one day an email to NASA to ask a question about their data, and then they proposed me to come for 1 year. And I said 1 year because I wanted to go back to Australia after, and that was 14 years ago. So yeah, that's how I got here. But obviously loving it, and it's a great place to work, and I feel very fortunate to be part of the agency and the team that we work with, and get the experience of the missions.
So sort of like Cecile and Jeremy, I was always interested in science. I was probably the only kid who dressed up as Galileo in elementary school for Halloween. But at some point, I actually got very interested in art as well. And I actually went to art school at first, photography school specifically, when I graduated from high school. And it was sort of a slow progression. I was in photography school. I liked images, and I got really interested in satellite images because, come on, they're just really cool, right? I got really interested in satellite images, and then how those images are formed and what they're telling us about the Earth. And so it was a sort of progression from art school to eventually getting my ultimate degree in applied math, technically. So one thing led to another. It certainly wasn't a straight pathway, but it went all over the place. And the one thing that was really, I learned over the years, and something I would recommend to other people who are interested in STEM, is to not pigeonhole yourself into I do this thing and this is my specific discipline. Particularly in earth sciences, but in other disciplines, other broader disciplines as well. There's a lot to be learned from other ways of approaching things. We didn't really talk about it that much today, but we benefit a lot from advances in the computer science world, specifically machine learning, artificial intelligence. A lot of this is being implemented in PACE, and it's such a new field. And so we brought on people from that kind of expertise. And so that sort of multidisciplinary approach, that sort of not pigeonholing yourself into one way of doing things or another, is so important to being successful at what we do.
It's so impressive to hear the work that all of you are doing, your mastery of this subject matter. Do you have any words of advice for any of our listeners who might be interested in pursuing a similar line of career or study in environmental science?
So I have three pieces of advice that I usually offer early career STEM researchers, and you probably won't like any of them. The first is to ensure that you diversify within the STEM field. Biologists, marine scientists, environmental scientists are all much, much better when they've taken a ton of math classes and a ton of physics and a ton of chemistry. You know, those hard sciences that really teach you how to move numbers around. Vice versa. All of those physicists need to take a few biology classes so they develop some empathy for this system that involves life and is nonlinear. And then all of the above, you need to learn to program. You just can't get around it. So that's advice number one, just suck it up. Two, write as much as you can. Write and practice writing. If you can't communicate on paper, you're not going to win proposals and get published, and you just have to do it. And the third, which most people hate the most, is any opportunity you get to be a public speaker, you take it. You need to be in front of an audience. You need to learn how to connect with the audience and communicate. Then everybody gets up there the first time and is terrified. You get up there the 100th time, you're terrified. I've been doing this for 25 years. There are still occasions when I get on stage and you can see my hand shaking. Again, you got to just muscle up and do it. So good luck.
Yeah, well, and I'm going to try to build on that with some other advice. And, I think we have heard in all three of how we got there. It's not a straight line. And I think, the advice I generally give is to be very open-minded, don't ever close doors. You don't, sometimes I have had stuff like that email that I wrote where I was like, it's going to get into someone's spam folder and I will never hear back. And it ended up being my job. That's where I ended up, that one email. So even if something seems crazy and no chance that it's going to work, try. The second piece of advice is to be strategic. That was one advice that I was giving as a PhD student. As an undergrad, actually, was to think, in 5-10 years from now, what do you see society is going to need? And try to align your skills with that. For me at the time, it was we would no longer be able to be a biologist or an engineer. You had to have a multitude of expertise. You couldn't be an expert in everything, but you should know a little bit more than just your field. And my last piece of advice is have fun. At the end, you're going to spend 8 hours a day at work, probably more. Don't do something that makes you feel miserable, because it's going to be it's hard to start with, but do something that you think is exciting and makes you really exciting and makes you think about it when you drive and stuff like that. So have fun, most importantly.
I wish we had more time to continue our conversation. This has absolutely been fascinating. My heartfelt thanks to our panelists for this topic of the Thought Leader Series, NASA's PACE mission, Unveiling Earth's Ocean, presented by UTMB. Please join us for future programs. And again, thank you so much for this fascinating discussion.
Thank you.
Thank you so much.
All right. Thanks, everyone.
Cheers all.
Decades in the making and launched mere months ago, the Plankton Aerosol Cloud and Ocean Ecosystem, or PACE, is giving us a first glimpse at promising new data from the sea and sky.
It's a mission that's exciting because PACE makes the invisible visible. It's flying hundreds of miles above Earth, and yet it's designed to see microscopic things.
PACE is seeing the ocean like no other mission has before. While previous ocean color satellites could only detect a handful of wavelengths, PACE is detecting more than 200 wavelengths. And more wavelengths mean more pigments.
Tiny organisms called phytoplankton contain green pigments that allow them to photosynthesize, which is really important if you care about breathing and eating. But their pigments make them many, many shades of green, and that contains a ton of information.
The more information revealed by PACE's hyperspectral ocean color instrument, the more details that emerge from our changing ocean and coastal communities, like how one group of phytoplankton can dominate a particular area.
You can see kind of that in chlorophyll, and you see that in chlorophyll, that pigment. But once it starts popping up from this view, the PACE allows us with all these different phytoplankton. Once again, you see so many different miniature, teeny tiny features that are maybe not that important physically, but they're important enough to make one type of phytoplankton win, one type of phytoplankton be dominant versus the other.
And you care about this because not every community is the same. Some are beneficial. They provide food and oxygen. Others close beaches. So we really want to know where they are and when they appear and the impact of all of this.
PACE is also giving us an early look into aerosols, microscopic atmospheric particles like dust, sea salt, pollen, and volcanic ash. The two polarimeter instruments on board measure the light bouncing off the particles at many different angles.
This scene represents observations at a number of different geometries as the spacecraft flies over. So it's about 5 minutes of time elapses between the first observation at one angle and the last observation looking in the backwards direction.
Moving through the different angles reveals the sun glint reflecting off the ocean surface. Analyzing the geometry of that sun glint gives us more details about that environment.
If you want to observe the ocean itself, inside the ocean body, you can't do it when you see the sun glint reflecting off like that. So having lots of different viewing angles allows you to see geometries where you can see what's happening in the body of the ocean as well.
That's only the surface. We really understand where the color comes from, what is really inside in the atmosphere, and what is inside the water, right? There's a deep meaning of those stuff. And so in order to really understand them, we need more information.
And I cannot wait to be on the far end of launch with data flowing and data quality looking great, just absorbing what the community comes out with, because I think we're all going to be really surprised and really, really pleased.